involves looking at small marine animals. Now, these animals are fascinating but often go unnoticed. So one of the first things we're going to show you is how to go out and collect them. A simple way to actually find these hidden creatures in the sea. We're then going to show you how to build some simple experiments to actually investigate something about these animals. And specifically, most of these have eyes, so how do they respond to light? And finally, we're going to show you how to build some simple microscopes. So these are really tiny creatures, and a simple microscope will allow you to get a much better close-up look at them. But these are the sort of things that we're going to show you here are the sort of things that you can use to answer all sorts of other questions. You don't just have to look at marine animals down a microscope. You can look at whatever you want. And we're just going to try and put these tools in your hand and show you how you can answer whatever questions you might have about the world around you. So when we're out collecting animals from the sea, we'd normally use something like this. And this is a specialized plankton net. It's basically just a big bag of mesh, which we pass through the water. The water gets filtered through, and anything that's in the water will collect at the end. So we've got this little filter at the end. We can open that up when we take it out and see what we've caught. The thing is, this is a pretty specialized piece of equipment, and we know not everyone's going to have something like this. But you don't need something as technical and specialized as this to actually see what's in the sea around you. So we've come up with a solution which we think will do basically the same thing, but is much easier for anybody to just make themselves.
All you need is a bottle of seawater and a packet of these. And what you're going to do is take one of the tights and put it over the top of the bottle like this. Then you go and push it all the way down, and then you're going to pour the bottle of seawater into a container so that everything that's inside gets filtered to the tight.
That's it.
So now that we have that emptied, we're going to very carefully peel this off and see what we have left behind in the tights. So, like so. Very cold.
Yeah,
now you can look at what's in the tray and see if you've caught anything interesting. So one of the animals that we perform experiments on here in our lab are called brine shrimp. Now brine shrimp are also called sea monkeys, and they're these tiny sea creatures that love salty water. And we can actually hatch them here in our lab, and it's also very easy for you guys to hatch them at home. Now, this is the system that we use to hatch brine shrimp here in our lab, and it's very simple. All it is a container in which we've put some water, a little bit of salt, and some brine shrimp eggs. Then what we do is we attach a piece of tubing to an air pump, and then we make sure that we have air flowing through this tank. And this essentially ensures that the water is always moving and that we have enough oxygen in the water for the brine shrimp to hatch. The other thing that we have here in this system is an LED light, and that's because the brine shrimp eggs need light in order to hatch. Now that we've shown you what the brine shrimp hatchery looks like in our lab, we're going to show you how you can make one of your own at home. To do that, you're going to gather a few things first. You are going to need 2 bottles, a pair of scissors, a drill or a Dremel, a pair of safety glasses, an air pump, a valve, an air stone, some silicone, and salt, water, and brine shrimp eggs. The first step in building a setup is to build a hatchery. And to do that, we're going to take 2 plastic bottles. The first thing we're going to do is we're going to take the lids off the plastic bottles and set them to the side. We're not going to throw them away because we're going to need these lids later. Then what we're going to do is we're going to take one of these plastic bottles and we're going to take our scissors and we're going to cut it in half. So the easiest way is to make a hole and then we're going to cut around this hole. This is going to be the base of our hatchery. The next thing we're going to do is take the second bottle and we're going to cut the base of it off. So to do that, we take our scissors and we cut around. This is going to be the hatchery itself. When I showed you the hatchery that we have built in our own lab, I showed you that we had air running through it. And that air is really important for two reasons. So first of all, we need movement within the container for the brine shrimp eggs to hatch, but also they need oxygen. And the same goes for the hatchery that we're going to build at home. So how are we going to do that? Well, the first thing we're going to need is an air pump. Now an air pump like this you can find in any pet shop or any aquarium shop. The second thing that we're going to need is a one-way valve. Now this is really important because we don't want any water getting into this pump. And one of these valves is going to make sure that that doesn't happen. Then the third thing that we need is one of these. This is an air stone. So this basically is a piece of stone that has loads of holes in it. And this essentially means that instead of having one big bubble going into the hatchery, we're going to have lots and lots of little bubbles. And that's really good because it means that we're going to get more oxygen into the water, and that's going to be really good for the brine shrimp. Okay, so how are we going to put this together? So the next thing we're going to do is we're going to take one of our bottle caps from earlier and we're going to drill a hole into the center of it using our drill. Now to do this, it's really important that we wear our safety glasses. Like that. So now that we've the hole drilled in our bottle cap, what we're going to do is take our air stone and poke it through. Like this. Boom. Done. Now the next thing that we have to do is we have to take a small piece of rubber tubing and fit it on over here like this until it's nice and tight. Next we need to make sure that this joint here is watertight. So to do that, we're going to use some silicone. So what we're going to do is apply a silicone in a ring all around the outside here. Next, we're going to leave this to cure for 12 hours. Take the base of the hatchery and drill a hole into the side here. Now that we've let the silicone harden for 12 hours, it's time to start putting the hatchery together. So first, we take the main body of the hatchery and screw the bottle cap on like so. We take the base of the hatchery and we're going to feed our rubber tubing through the hole that we drilled earlier, like so. Then take our one-way valve and we're going to fix it onto the end of the rubber tubing, like so. Then we take our air pump and our hatchery and we're going to attach the end of the rubber tubing onto the other end of the one-way valve. like so. Now that we have our hatchery assembled, it's time to start filling it up. So the first thing we do is fill up our hatchery with water. Now the water that we're using for this is just plain tap water. Now brine shrimp love salty water, and tap water is not going to be salty enough for them. Here in the lab, we use special aquarium salt, but you guys at home can use normal table salt. Now what you're going to need is roughly about 4 teaspoons of salt per litre of water, and if you put in a little bit extra, it's absolutely fine because the brine shrimp love it salty. Add your salt to your water and it's going to sink to the bottom and then slowly start to dissolve. Now when you buy the brine shrimp eggs, they're going to come with their own instructions, and that will tell you how much to add to your system. For what we have here, about 1 scoop is going to be enough. Okay, so when you first add the eggs, they're going to float on the top and then slowly sink down. And it's at that point that we want to turn on the pump. And you can see that we start to get bubbles. This is really good. Now we want to leave our hatchery somewhere where there's a lot of bright light, and we're going to leave it for roughly a day to 2 days to allow the brine shrimp to hatch.
So the next thing we're going to need for our experiment is to be able to control some lights, and then we can record how our animals will respond to these lights. So the lights we're going to be using are these pixel rings. You can get them in different sizes. This is 16 pixels and this is 12 pixels. And to control them, we're going to be using an Arduino. This Arduino will send signals to the lights and switch them on or off, and also change the color. And the last thing we're going to need is we want to be able to control these lights ourselves. So we're going to wire up a Nintendo Wii nunchuck, and this will be able to turn the lights on and off, decide which pixels are on, and change the color. So first I'm going to show you how to wire up the pixel ring to the Arduino. So the pixel ring has 3 pins. It has a 5-volt pin, a ground pin, and a data pin or digital pin. So the Arduino also has a 5-volt, ground, and digital pins here. So we're going to put the 5-volt pin into the 5-volt on the Arduino, the ground pin into the ground pin on the Arduino, and the digital pin into digital pin 6 on the Arduino. Could be any pin, but in the code we have specified it goes into digital pin 6. So next we're going to plug in our Arduino into the laptop to give it power, and we're going to run one of the demo files for the pixel ring, and it's called strand test. So now you can see the pixel ring is lighting up in all different colors. So in the panel on the bottom we can see the signal coming from the sensors in the nunchuck. In the top left hand corner we can see the code that we've used to program the nunchuck. So each of those lines on the bottom there represents a movement of the nunchuck. So if we tilt the nunchuck side to side, we see the green line moves. If we tilt the nunchuck up and down, we see the yellow line moves. And then when we move the joystick on the nunchuck side to side, we see the blue line moves. And up and down, the red line moves. So using this, we can control the lights on our pixel ring.
So we've set up this rig, we've done all the coding, we've got all the wires connected, and we've got it looking nice and neat. But now we need to actually do the experiment. We need to find out, are these brine shrimp going to react to light? So we take the little brine shrimp, put them into a clear jar, and place them on top of your little NeoPixel ring. So the light can basically come at them from any angle we decide to. Now, when you're doing this experiment, we want to make sure that we get rid of any factor that could affect the results of it. So if we're testing, are these animals going to respond to light, we need to make sure that this is the only source of light in the room. So when you start this experiment, just make sure you turn off the lights. So we've got the NeoPixel hooked up, we've got the nunchuck hooked up so that we can use it to control the light. And now what there's left to do is to do the experiment. So leave it dark at the start, let the animals get used to their environment. So they're getting used to this lack of light. After a few minutes, they'll be used to it enough, and we can then take the next steps of actually starting the experiment. So make sure you've got your hypothesis in your mind. What's your hypothesis in this situation? Do you think they're going to respond to light or not? Let's apply some light in one direction. Now, there's some important questions to ask yourself when you're doing this. Are all the animals moving in the same direction? Are some of them moving in other directions? Have we controlled for every variable that we possibly could? Is there anything else that could be affecting the experiment? Every time we do a scientific experiment, we're also always trying to answer a question. In this case, will brine shrimp respond to light? Will they move towards it? Will they move away from it? Or will they have no effect whatsoever? So when you're doing an experiment like this, it's really important to bear in mind, biology is not clear cut. There's never a nice, simple yes-no answer. It's always degrees of variability. And animals, like ourselves, like brine shrimp, always have a degree of variability in them. So you should see in your animals that they're moving towards the light, or at least most of them are. Not all of them. Some animals move to the other side of the ring. Some of them move randomly and don't seem to follow the light at all. That's because animals are variable. They're not meant to move, or they won't all move identically to one another. Now, we know brine shrimp in general respond to light. They like to move towards it. So we've answered that question. But we need to make sure we've got the right answer here. And in science, what we do is called repeats. We repeat the experiment start to finish again, a couple times if we can, and see, do we find the same answers that we did the first time? And if we do, then it means, as long as our experiment was run correctly, that we've got an answer that we can be fairly certain of. We can be confident in our answer. So now we need to think about why that is. Why would brine shrimp move towards the light? Most animals will move or live and interact with their worlds in a way that provides them with the things they need to survive. And the biggest example of that is food. So, if you've got an animal that feeds on, let's say, plant matter, we know it's going to want to be places where there's more sun, because plants, we know, like the sun. And if we follow the sun, then we're going to find plants to eat. If you feed on the smaller animals that feed on those plants, you're going to have the same principle. You're going to be trying to follow the sun, follow the light, so you can find the animals that are eating the plants. Now, there's a bunch of other reasons that could be at play. It's going to be lighter, more light is going to be available towards the top of the water. Maybe there's something special about the top of the water that we haven't thought about. We haven't tested for that in this experiment, but maybe you could figure out a way of testing for that yourselves, using the skills that we've just learned here.
So one of the things we do here as part of our research is we build a lot of our own equipment, and in particular we build quite a lot of our own microscopes. Now the microscopes that we use for research can be relatively sophisticated, and they might not necessarily be the sort of thing you're going to be building at home. But we do have a couple of little tricks to make really quite useful microscopes much more simply. And the first of these I'm going to show you, and this is one of our favorites, is to basically modify a webcam to allow it to basically function as a microscope. We have a favorite webcam that we use for this, and this is a Logitech C270. It's relatively cheap. You can find it from lots of different places. You can buy it online and whatever. It's not the most powerful webcam in the world. It's not the most highest resolution or the best quality or anything like that. But what it is one of the easiest to modify and hack and turn it into, in our case, turn it into a microscope. Now, when you buy one of these webcams, there's a plate mounted on the front, which you can just unscrew. I'd show it to you, except years ago I lost it, so you have to use your imagination. There are 3 screws which basically hold it in place, and we've actually replaced it here with a little 3D printed part. There are 3 screws which hold it in place, which I've removed, and then if you take that off, you can see the actual device itself. So inside here we've got in the middle there's a microphone. We're not using the microphone, we can ignore that. Some of the processor chip and so on. At the other end here there's a little power indicator LED. That can get in the way when you're using it as a microscope or using it really close up, especially if you don't have anything on the front. So a little bit of blue tack just to cover that up will solve any problems that light might cause to your sort of microscopic imaging. But the other thing that you're going to see here is the lens. Now when you get this camera, the lens is in a fixed position, and it's in exactly the position where you would want it for a webcam. So that lens is set up in front of the sensor inside this at just the right distance, so that something that is a normal distance away from the camera will just be in focus. So if you think about how we normally use a webcam, whether it's for Zoom or whether it's for whatever you're using it for, you want to sit in front of it and be in focus. That's great, but that's not a microscope. But that's where this particular webcam gets quite useful. Because when you take the casing off, the lens inside, which sits onto the sensor, is basically screwed into place. And what you can do, if you take the cover off, like we have here, is you can basically change this, you can unscrew the lens. And what this does is, so you can unscrew it and take it the whole way out if you want. That's what it looks like, that's the sensor inside. But what this actually allows you to do is to move the lens further away from the sensor. And when you move the lens further away from the sensor, then if you're using it as normal, if you're pointing it at your face, you'll be completely blurry. Because it's not focused on you, it's focused on something much, much closer. So the further away from the sensor you move the lens, the closer to the lens is going to be in focus. So we can use this to basically focus on something which is very, very close up. And if it's in focus and very close up, then it's hugely magnified. And that basically is what a microscope is. So when you're using a little simple microscope like this, one of the things that's going to make it much easier to use is to have a little stand for it. And what a stand will allow you to do is to hold it at a fixed height above your sample. And that's going to hold it stable and make it much easier to actually work with. So what can you use to make a simple stand? And you can make it out of anything. And what I find actually is really useful is Lego. So the thing about Lego is it's a very well-defined distance. So there's a single Lego block, a Lego brick, and there's also Lego plates. And 3 Lego plates together are the same thickness as 1 Lego brick. Okay, so let's be a little bit more precise about this. A Lego brick is 6.3 millimeters tall. The plate then, which is 1/3 the height of that, is 2.1 millimeters. So if we have one Lego brick plus 2 plates on top, the total height then will just be a bit over 1 centimetre. And 1 centimetre is about the right distance for a microscope like this. So if you have a sample about 1 centimetre away from the lens, that's going to give you pretty good field of view and should be pretty easy to focus in on something like that. So you don't have to use Lego, but it's an easy way to get a nice stable base. Lego is not a toy. This is serious engineering. Who am I kidding? It's a toy and I'm a grown man who plays with toys. What can I say? Okay, so just a little simple platform. And then we're going to pass through a cable tie here in the gap that we made here. Pop this on top. And this cable tie will just hold everything in place. So there you have it. A nice simple platform to hold your microscope. It's not the most stable thing in the world, it's a bit wobbly, but because it's Lego, all you need to do is just add whatever extra little bits you want to give you something much more robust and stable, and then that'll allow you to look at any sample you want, give you a nice close-up view of anything that you want. And just to, I guess, finish off, keep in the theme, we'll add a little diver there, and sure, why not, have a Lego shark. Rawr.
So microscopes don't have to be the big, fancy, expensive looking things that you see on TV. They can be really cheap. The only thing you need for a microscope is a lens. A lens is just a curved surface made of a transparent material that is used to bend the light in a way that it normally wouldn't go. What this does is it means that the things that are close up to the lens appear bigger on the other side of the lens. That just makes it look bigger. You can buy really fancy, expensive looking microscope lenses with lots of mini lenses in the middle of them. But we don't actually need that just to be able to see things close up. We can actually just use any old camera, and every single phone nowadays has a camera attached. So today we're going to go through, now we're going to go through how to turn your phone into a microscope. Now, we are going to be using a clear silicone to be able to generate these lenses. This silicone is pretty expensive, but there's no reason that other clear drying silicones shouldn't work. The only thing that you need to have a lens is a curved surface and a transparent material. So this silicone here is mixed in a 10:1 ratio. So if you add 10 milliliters of part A and then add 1 milliliter of part B, you end up with a silicone that cures really nicely. So there's 2 milliliters of part A. We only need a tiny little bit of silicone to make this work. And then we're going to add just 0.2 milliliters, just a tiny amount of part B. Okay. Because the silicone is made of 2 different parts, it means that we have to be really careful and mix it super thoroughly. If we don't mix it thoroughly, it's not going to end up setting, or you're going to end up with a mixture that's partly set in some places and partly liquid in other places. So go ahead and take a coffee stirrer and mix together the 2 parts. Keep mixing until it's all completely uniform. Now you'll see after mixing a silicone, you see all those little air bubbles in it. All those air bubbles are going to be a problem if we try and actually use this as a lens, because they're going to get in the way of the light and they're going to make it react in really strange ways. We don't want that. The way to deal with that is just leave it for a little bit. So silicone is really sticky and it's going to try and stick to everything that it touches. So when you're working with silicone, you're going to want to wear gloves. Now, latex gloves or nitrile gloves are going to be the best because they're going to let your hands move as much as you want. But if all you have is rubber gloves or any other kind of glove that's sealed and you can bin afterwards, that'll be fine. Now, if you're doing this in somewhere like a kitchen or in a surface that you don't want to get dirty, you're going to want to put down some kind of covering to protect that surface, because silicone, once it sticks into something, it's really hard to get out. Also, don't wear nice clothes while you're doing this. If silicone is going to get into your clothes, you're not going to be able to wash it out. Once your silicone's got all the air bubbles out of it from just resting it for a little while, we're now going to transfer that into small little blobs so we can make our lenses. So we want to take a small little blob of silicone and transfer it onto our flat surface that we need to form the lens. So just take a regular old coffee stirrer, scoop up a tiny little blob of the silicone, and then just press it in lightly onto your flat surface. When you pull away gently, you'll see that there's a nice little circular bubble formed of silicone. Do that with all the silicone you have. You can make any size you want here. Just bear in mind the size of the camera on your phone. Now, the more silicone we put onto these lenses, the heavier they're going to be, and that's going to change the shape of the lens when we do the next step of turning them upside down. So, I've got a few lenses of different sizes on there. Now what we're going to do is take whatever you've got your silicone lenses plated onto, and you're just going to flip it upside down. So the really nice thing about using Petri dishes like this is that they have this rim around the outside. That means that when we turn it upside down, it's able to rest on the floor or on the surface that you're leaving it on without the silicone touching anything. We don't want that silicone to touch anything other than just this nice little flat surface on the back of the Petri dish. The reason that we leave it upside down is because gravity is going to pull the silicone. The silicone is really thick, so it's not going to actually drop down as long as you haven't got too big droplets. But the gravity is going to pull the silicone down. That's going to create a really nice curve. It's that curve that we need to make the lens. So this silicone takes about 12 hours to cure, so we're going to leave that overnight, just at room temperature, somewhere safe where it can't accidentally drop down and damage something underneath it. And we'll come back tomorrow and see how they look. You're going to have to bring it quite close to the camera to be able to see. and you'll see all this fine detail, way more than your phone would have been able to capture before. Now it's a little bit blurry at parts. That's because the lenses that we're using are not perfect. Here we have some sodium chloride. It's just regular old table salt. Just going to spread that onto a little dark surface. Now if we have a look with our new phone microscope and zoom in, what look at first like little balls actually turn out to be really neatly defined crystals. See all those solid cube shapes? You have to go really, really close and focus on that little bit in the center. That's going to be where you're going to find the clearest picture.
So hopefully we've been able to show you a couple of tricks which will allow you to go out and explore the world around you, to look at the world and ask questions about it and get those answers. And if you do come up with anything interesting, be sure to let us know. Thanks very much, folks.
That was cool. Yeah, so that was cool. Thank you for that. So I noticed that live chat has been quite busy. So if anyone have any questions, you can keep asking them on live chat. I haven't been keeping track on all the kind of. Have you answered a lot of the questions on live chat? So Laura, you might be able to jump in there as well.
Yeah, I have one. What's the difference between table salt and aquarium salt?
George, do you want to answer this? Because I don't know what the aquarium salt is.
Oh, sorry, I thought you said aquarium salt. The salt you use at the
Yeah,
beginning.
no, it is aquarium salt. George looks after the aquarium. I just don't know what it is.
Yeah, so the thing about the aquarium salt is that I don't know what it is either. They
Magic.
don't
Okay.
list what's actually in it. So they'll usually just sort of say, oh, add this much and you'll get this salinity. But they won't give you the details of their sort of special secret formula. Which works for some things, not when we're looking after the actual big jellies, because we need to be really, really precise about exactly what they get.
Okay.
Yeah, I'm just looking through live chat at the moment. Yeah, I think people are pretty impressed with the silicon lens. I was as well when I first saw. I was like, oh my god, that's pretty cool.
They're great, and the thing is, everyone is different, right? Because it's all just a drop of silicone. So if you make up a batch, some of them will be rubbish, some of them will be higher magnification, some of them will be more distorted. So it's great fun to experiment with. You can just sort of play around with them, and they're really easy. I guess the only problem with them is the specific silicone. So the stuff that we use is pretty specialized and can be hard to get your hands on. But there's tons and tons of different silicone out there on the market. We haven't tested them all. We've literally just tried two things. There's the Silgard stuff that we normally use, and then we just use plumber's silicone. And the plumber's sealant stuff did not work at all. That was a disaster.
That's good to know, because that's probably like first thing if the hardware stores are open.
Yeah.
Whenever I suppose it's kind of like when you say like a sealant, kind of silicon sealant, that's the first thing that comes to mind is like, go to the hardware store and buy one of those. But that didn't quite work.
No, it doesn't work. I think it's just it's not runny enough. It needs to be more liquid. It needs to be a bit runnier and it's a bit too stiff. If you could work out some way there's probably something you could add to it to soften it or make it more fluid, but I know nothing about silicone chemistry. So I have no idea how you would do that. But there's a huge range of silicones available on the market. Smooth On is one company I'm aware of that sell a vast range, and they're used a lot in things like makeup and things like that. So, and they all have cool names like Dragon Skin and stuff like that. Yeah.
So if people who are not close to see, I know in the shopping list you mentioned that people can buy them in. You mentioned one place is Steers Aquariums, I suppose.
Yeah. So
is there any other places like any kind of pet store will have them as well, or
the Brine Shrimp?
Because I know they're based in Ballymount. I used to go there because we used to have fish, but it's not kind of easily accessible for people.
It's not. No, it's not easy. You can buy. So brine shrimp are usually used as food in aquariums. So people hatch them basically just to feed to their pets, which is the grim side of pet keeping. I guess if you have a cat or a dog, I guess you don't have to do the actual, tend not to have to feed them live animals, so it seems a little less gruesome. But most pet shops will have them, and lots of pet shops you can buy online. You can probably buy them online from I'm pretty sure you can buy it from places like Amazon and stuff like that if you want as well. But most local pet shops will sell them. We actually ran out of, and actually, the one thing is, if some of them will actually sell the live ones as well. So I believe Seahorse will sell you a bag of brine shrimp that have already been hatched if you don't want to go do the hatching. But sure, that's the fun part.
Yeah, so just to let people know that Seahorse Aquarium, they're the ones based in Ballymount, isn't it?
Yeah.
I think there's a question from Wild Dairyman. Any experiments with black lights on marine life?
No, we haven't. So I guess sort of black light, it tends to be, that's kind of the ultraviolet end. And the thing about ultraviolet light is it doesn't penetrate the sea very well. So so this is the thing, and one of the things that's kind of, if anyone does go and play with this, a really interesting thing to do is in the version of the experiment we just showed white light. But it's really interesting to compare different wavelengths of light. So blue light, green light, and red light, and infrared and ultraviolet if you happen to have it, which you probably don't. But so the thing is that so animals can tell color, they can sense color. Lots of animals that live in the sea can sense color. But they're maybe not seeing a color image. What they're probably doing is they're using the color to judge depth, because the red light will penetrate much deeper into the water and the blue light stays much closer to the surface. So, if you can detect blue light, then you're very close to the surface. So a lot of animals, so actually if you do this experiment with brine shrimp or with copepods, they're very reactive to blue light and much less reactive to red light, because blue light's about the surface. So ultraviolet light is even shorter wavelength than the visible wavelength that we see. So wouldn't penetrate the sea very far at all. But so I'm not sure. Actually, it'd be really interesting to sort of to see what happens. And one of the other things that we do is we've got a couple of small remotely operated vehicles, basically underwater drones that we can stick cameras on. We stick cameras on and stick them in the sea and see what we can find. And one of the things that we're kind of interested in from a research point of view is looking at the behavior of animals in the wild. And it might be interesting to kind of stick on some, because we do have some ultraviolet and near ultraviolet light. Might be interesting to stick on and see how animals react to it and respond to it.
I can attest to that. I worked on a project with blue lights on horses, so it'd be interesting to see under the water, because as you say, not all the light can penetrate equally. It'll be at different depths.
Yeah,
that's why I was so surprised to see that the jellyfish went to the light. I thought they might have an aversion to light being underwater.
Yeah, well, this is the thing, because a lot of animals will. So, I mean, of the stuff that we kind of hauled out of the harbour in Wicklow, we got a couple of small shrimp and crabs and stuff like that, and they tend to kind of sit around the seafloor. So they don't have a particular. They can see. They can definitely. I mean, crabs have big, complex eyes, and they can definitely see. And actually, they will if we go near them with the ROV, they will actually get defensive, which is always kind of funny, because you get a crab which is maybe about 10 centimeters across, and you drive up to it with a big ROV, and they kind of get themselves up. Right? They get into this defensive posture where they kind of raise themselves up on their legs and put up their pincers as if to kind of go, try it on, so they can see it. But they don't seem to be that. It doesn't really. Light by itself doesn't seem to produce much of a response to them, but that's because they live on the seafloor, so it's not a big deal for them. For other animals that live in the water column, like the little tiny copepods, which are basically a little tiny shrimp, or the brine shrimp, which we hatch ourselves, they're going to basically they need to go to the surface, and they like to hang around the surface, largely because that's where their food is. So that's why they tend to go around there. So they will mainly eat things like phytoplankton, which is tiny microscopic algae. Yeah, so I've lost my train of thought. Sorry.
I have another question. From South Dublin Radio Club regarding phosphorescence in sea creatures. Are there any perceived future applications that can be derived, for example, medical diagnostics, etc.?
Well, it's really funny because I don't know necessarily. Well, I'm trying to think now. Well, the first sort of fluorescent protein. So, a lot in kind of modern day lab biology, people deal with fluorescent proteins all the time, and we can get the gene for a fluorescent protein and stick it into a cell and make that sort of cell fluoresce. And that's a kind of a fairly routine thing to do now in kind of biological research. And that's the original fluorescence protein was identified in jellyfish. So there's a lot of fluorescence that happens in the sea. So people have actually, for quite a long time, been kind of working out how fluorescence and how light works in marine animals and actually harvesting that and using it for other applications, mainly research applications rather than kind of, you know, something you might put in a human. We tend not to try not to make people fluorescent most of the time. The luminescence thing is different. So that's, so, you know, fluorescence, and maybe lots of people don't necessarily know the distinction between them, but fluorescence is where you take in one color of light and you emit another. So you need to put light into a system to get some fluorescence back out. But luminescence, and lots of marine animals are bioluminescent. That basically, it generates its own light. And some of those, so usually what happens there is there's an enzyme. So it's somewhere inside a cell in an animal is an enzyme. And when that enzyme becomes active, it's sort of usually it's they actually consume oxygen in the process, and then it emits light. So it consumes a bit of energy and emits a bit of light. But there's actually lots of again, the genes for that have been discovered. It's the same thing, actually. Particularly things like fireflies use a very similar kind of thing. The gene's been discovered, and actually that's used routinely in sort of molecular and cell biology. So, you know, you have a chemical reaction, you want to see it happening, but you tag on these luminescent molecules, and then you measure the light coming off, and then you can measure the amount of protein or enzyme or whatever it happens to be. So there is actually lots of use of these. So there's a lot of interest in kind of identifying these things in the wild and seeing how they work.
So, is there any more questions? Live chat. I think while Dairyman was just following up with some comments from his earlier question. I'll just read it out for people. It says, I filmed a lot of this behavior in inshore waters, including Portuguese man-o-war, amphipods, various fish, crabs, etc. So that's just follow-on comments. I don't think there's any more questions. Laura, do you have any more questions?
No, I think that's all it. I think Mark and Vincent from Wild Dairyland should follow up with each other, because I think you do a lot of very similar things.
Definitely, yes.
You'd have a lot to talk about, because I've seen Vincent's work and it's amazing. And it put me in mind of you with your seal. Actually, that's another thing. Your video on Twitter of the seal with the ROV.
Yeah.
So everyone should go check that out.
That was Amy actually who captured that footage.
Fair play, Amy.
Pretty cool.
Coolest thing I've done so far.
Very exciting, and I think she nearly jumped into the sea after it.
I got very excited.
Yeah.
So, kind of any last words, kind of announcements, or what you're doing, upcoming stuff?
No, nothing. I guess the only thing I would say is, you know, if anyone does do anything that, you know, use any of the things that we've shown here, like, get in touch and show us what you've done. I'd love to know. I'd love to see what people have done. That'd be great.
So I'm sure Laura feels the same. That thank you so much to you, Mark, Amy, George, and of course Niamh couldn't join us today. So thank you to everyone for making that video. It was really interesting. But yeah, totally. If people still have answers and any questions, you're on Twitter. And I've been tweeting, and how am I forgetting your handle? Pickering Lab UCD. Yeah. So on Twitter. So if any more questions, ping them there. But yeah, so what I'm gonna do now is I'm gonna wrap up now. I'm gonna say thanks to everyone, and also to Laura, And also mention as thanks to SFI, Science Foundation Ireland, because they also fund Dublin Maker, as well as Science Week. So I'm gonna say thank you all, and hopefully we get to see each other in person in the near future. If not, let's do something again, even virtually. So I'll announce the next event for tomorrow. So thanks again, everyone.
Bye.
Bye.
So again, thank you to Jellylabs, the whole team there. So tomorrow we'll be back at 1 o'clock with Fab Lab Maker Hub, Stuart Lawn, who's going to be showing you Lego modeling in Tinkercad. So don't forget to subscribe to our channel and hit that notification bell so you get to find out when our next video goes live. So we're on social media. We're Dublin Maker on Twitter, Facebook, Instagram. And so we'll see you back here tomorrow live. Bye, everyone.